Good evening, ladies and gentlemen. Welcome to the Crick Lecture, 2011 Crick Lecture, or is it 2012? Not sure which one it is. Anyway, to the Crick Lecture. Before we start, could I ask you to turn off your mobile phones because this is being webcast and recorded. Thank you. I'm Jean Thomas, and I'm the Biological Secretary of the Royal Society, and it's a great pleasure to introduce this evening's speaker, Simon Boulton. So the Crick Lecture is given annually in any field of the biological sciences, but preference is given to genetics, molecular biology, and neurobiology, which are the general areas in which Francis Crick, who is a fellow of the Society, as you know, worked, and also to fundamental theoretical work, which was the hallmark of Crick's science. The lectureship was endowed by his longtime friend and colleague, Sidney Brenner, Fellow of the Royal Society, the first lecture being given in 2003. So this year's recipient is Dr. Simon Boulton, who's a senior research scientist and group leader in the DNA Damage Response Laboratory at Cancer Research UK, London Research Institute, Clare Hall Laboratories, which is quite a mouthful. And he's also an honorary professor at University College London. Simon was an undergraduate in Edinburgh and then did his graduate work with Steve Jackson at the Gurdon Institute in Cambridge. There followed two postdocs covering about 4.5 years in the US at Dana-Farber Cancer Institute and Harvard Medical School, and then at before that at the Mass General Hospital Cancer Center with Mark Vidal and Nick Dyson, respectively. At the end of all of this, as a result of what he learned during his PhD and his postdoc, he was firmly hooked on studying the DNA damage response process, and that's what he's going to tell us about tonight. Since joining Clare Hall in 2002, so it's almost 10 years, Simon's earned a reputation for being a leader in the field of DNA repair, DNA damage checkpoint signaling in meiosis. He chose to develop Caenorhabditis elegans, the worm, as a model system for the study of DNA damage response genes. There was very little published at the time, but he was convinced when he started on this that the power of genetic approaches in the worm would allow him to make significant contributions in a relatively short time and to cast a niche for himself. And he's proved right in this, and C. elegans has formed the basis for much of his work since 1992. So the lab currently exploits the respective experimental strengths of the worm, C. elegans, cultured mammalian cells, mouse models, and biochemistry to investigate the DNA damage response. And it's this combination of approaches that's led Simon Boulton's laboratory to make a number of seminal contributions to the field of genome instability in cancer. So, ladies and gentlemen, my great pleasure to present to you Dr. Simon Boulton, this year's Crick Lecturer.
Thank you very much, Jean. It's a great honour to be here. We'll start at the first slide, I think. So what I'm going to tell you about today is going to give you an introduction to what I've been very much interested in for nearly 20 years, which is genome instability and how genome instability is dealt with. And this is a good indication of what I'm going to go on and tell you about. Of course, this is the Crick Lecture, and I think it's appropriate to start this lecture by showing arguably one of the most famous pictures in biology, which is, of course, Watson and Crick with their structure of DNA. And this is, of course, the X-ray diffraction pattern, which led to this arguably the most important discovery in biology. So DNA, of course, is the blueprint of life, you could call it, essentially. It's made up of 4 different types of bases, and the sequence of those bases provides the genetic information for life, essentially. So we have 4 bases, A, G, T, and C. And, of course, DNA is a double helix, and so these bases pair with a cognate base, A with T and G with C. And it is this that really defines the coding and information within DNA. Now, of course, the DNA structure is extremely well accepted, but there is one problem with DNA, and that is it's intrinsically unstable. It is prone to chemical attack, modifications, breakage, as you will see. And of course, this is inherent. It has to be. If DNA was completely inert, there would be no option for change and for the selection of genetic diversity. So DNA has to be unstable to an extent. And this is really what we've been studying. So it turns out that DNA can be modified, it can be changed in many different ways, and this is just an overview of the different kinds of changes that can happen. The very act of metabolism generates reactive species that can interact and genetically modify DNA. You can have reactions with DNA as a result, and these change the coding information within DNA. You can have discontinuities in one helix, which would be a single strand break, or you can have a complete breakage of both of the helices, which is a double strand break. So this is a major problem, of course, because if you can't correct these different types of DNA damage, then you're going to accumulate mutations. This will accelerate aging, and this can lead to cancer and other genetic disorders. So how does a cell deal with DNA damage? Well, cells have evolved a variety of different DNA repair mechanisms. They do exactly what I've just said. They repair these DNA damages. So we have a variety of different repair mechanisms that correct the different types of DNA damage that can occur. And what I'm going to do is just hopefully give you an indication of the importance of DNA repair and how this impacts on human disease by focusing on one particular example before telling you a little bit about our work. And that is the types of DNA damage that are induced by sunlight, UV and sunlight. And I'm sure everybody is aware of that, that UV sunburn can lead to cancer. The question is how? Well, it turns out that UV light, as you'll see, induces a change in the DNA, and this is ultimately needs to be repaired to avoid mutation and cancer. So this is a man. He's lying in a park. He's obviously exposed to sunlight, and we're going to focus in on his hand. We zoom in a little bit. You can see a close-up of his hand. We move into the skin and the keratin, which is the proteins that make up the skin. We go a little bit deeper. Eventually, you'll get to a cell, and we're made up of millions and millions of cells. We then get through to chromosomes, and these are essentially the units that DNA is packaged into in our cells. This is a single chromosome, and you can start to see the DNA strands within this complex structure. And eventually, we get to the DNA strand. So we've zoomed into this man's hand. He's been exposed to UV light, and it turns out, and this is just one of the types of DNA damage that can be induced by UV, is that thymines are adjacent to one another. In the energy of UV light can cause them to dimerize. So this looks pretty innocuous, this slight change. But this changes the coding properties of the DNA. If you can't repair this correctly, this can lead to mutation. And of course, mutations lead to cancer. And this is very clearly illustrated by a rare disorder called Xeroderma Pigmentosum, which is completely defective for the repair of UV-induced DNA damage. And these children, unfortunately, are extremely photosensitive, and they develop skin cancers at a very high frequency. Now, fortunately, we can catch this early, and they can be protected from sunlight. But still, this really has been a very important paradigm studying this particular disorder, and the understanding of this disorder has really placed the repair of UV damage in context. So that's UV damage. What I'm going to tell you about is what I'm interested in, and that is arguably a more catastrophic DNA lesion, and that is the double strand break, where both strands of the DNA helix are broken. So one way that double-strand breaks are generated in DNA is by ionizing radiation. So we use this, of course, in cancer treatment, radiotherapy. So ionizing radiation can induce breakage of both strands of the duplex. And we can study this in cells by various means. One way is actually to visualize the double-strand break, and we know that there are various repair factors, damage response factors that are recruited. Sorry, it's gone a bit quickly. That are recruited to the double-strand break, and one protein that we're particularly interested in my lab is 53BP1, and this protein, as you can see, it coats the DNA double-strand break, and you can actually see these foci, and these correspond to sites of double-strand break in the cells. So we can study the dynamics of this and how these proteins actually behave. So I've mentioned ionizing radiation as a source of spontaneous double-strand breaks. And in fact, calculations have been done that on average, every cell in your body each day has about 10 double-strand breaks. And we have to repair those accurately. But actually, the major source of double-strand breaks in our cells is actually as a result of collapse of replication forks. So what does that mean? Well, replication is the process through which we copy our DNA. And generally, this is very accurate, but occasionally the machinery that copies DNA stalls and sometimes collapses, and this can give rise to double-strand breaks. It turns out, though, that our cells actually use double-strand breaks to trigger various processes, and these are called programmed double-strand breaks. And actually, our immune system is absolutely dependent on programmed double-strand breaks. If you don't utilize these breaks, you do not develop an immune repertoire. None of us would be here if it wasn't for meiotic recombination. So this is absolutely essential for the generation of gametes for sexual reproduction. And this whole process is absolutely dependent on a programmed double-strand break that prevents, or at least helps to ensure that chromosomes correctly segregate during meiosis. There is also another class of double-strand breaks, and these are actually what we call masked double-strand breaks. So telomeres, and I will come on to these are the ends of linear chromosomes. Of course, when your DNA ends, you have a double-strand break. And this, of course, would be a problem for linear chromosomes. So we have these specialized structures at the end of linear chromosomes, telomeres, which act to mask the double-strand break from being repaired, essentially. So why is this important? Well, defects in the repair of these lesions, whether it's ionizing radiation or collapsed replication forks or the generation and repair of these programmed events, give rise to a multitude of genetic disorders in humans, and many of which give rise to accelerated aging or cancer predisposition. So this is a very simplified view of how you repair a double-strand break, and it's basically there are two major processes depending on whether or not you've replicated your DNA, that is, copied it. So this is a very simple view of the cell cycle. This is the process through which DNA is essentially copied and then ultimately segregated. So in the gap phase 1 of the cell cycle, you only have one copy of the DNA, but once you replicated it during S phase or synthesis phase, then you have 2 copies. And depending on whether or not you have 1 or 2, really dictates how you repair DNA damage. So this is a very simple view. If you get a double-strand break when you only have 1 copy, the only reasonable way that you can repair this double-strand break is a gluing mechanism, essentially sticking the break back together. And we know many of the factors that are involved in this. The problem with this process is it's intrinsically error-prone. So a double-strand break generated by ionizing radiation, the bases at the end of the double-strand break are often damaged. You have to remove those bases before you can stick the ends back together. Of course, you're going to lose genetic information if you do that. Also, of course, if you the double-strand breaks that are repaired by this gluing mechanism, non-homologous end joining, they don't actually require sequence information. So breaks from different parts of the genome can actually be rejoined, and this actually drives translocations, which you can see here. So this is the result of error-prone repair by non-homologous end joining. You can see here a chromosome that has 3 colors. This is actually a chromosome made up of a translocation between chromosome 17 in purple, chromosome 16 in orange or yellow, and blue is the X. So this is the consequence, is as a result of inappropriate repair of double-strand breaks by non-homologous end joining. And this is a major driver in cancer. Okay, so what happens once you've copied your DNA? Well, if you get a double-strand break in one of these copies, you have an alternative option for repair, and that is to use an accurate repair mechanism of repair, which is called homologous recombination. So these two DNA duplexes are identical, so you can actually use the sequence information here as a template for the repair of this break. So you can use this using homologous recombination to repair the break, and I'm going to go through some of the details of how you do that. So essentially what you need to do is the following. So if you've got a broken end in one chromosome, you have an identical, what's called sister chromosome, which you can use as a repair template. So what you can see here already is one of the things that you need to do to activate recombination is to process the break. You chop it back, you chew it back, and I'll go into the details of this in a minute. You process this back, and this is a substrate for invasion of this broken end into the intact template sequence. So this is what happens. It invades. You then copy the genetic information that is missing, and you're left with this structure. This is a very famous structure. It's called the Holiday Junction. And through a bit of magic, you process this and you repair the break. Okay. So how do you do that? Well, it turns out that one of the major players in recombination that actually catalyzes that invasion event is a protein called Ra51. That's what it's called in you and I. In E. coli, in bacteria, it's called REC-A. Essentially, it does exactly the same thing in bacteria and in you and I. So basically what these proteins do is that they form these striated filaments on DNA. So they can bind to both single and double-stranded DNA. And there's a lot of information about this gathered over a number of decades, and we have crystal structures of these, and we know essentially how this filament works. So what this filament does is it binds to that processed double-strand break. It forms this filament, and this can then search for and catalyze invasion into this homologous sister chromatid or sister chromosome. So it can form what's called a synaptic complex, where the broken DNA molecule has begun to be engaged with the intact homologous sequence. This then undergoes a transition where the invaded strand pairs with its complementary sequence, and the non-complementary strand is displaced. And this forms a displacement loop or a D-loop. So this is part of the process. And of course, this now is a very nice substrate for replication. So you can then prime copying of this strand here, and then you can use that to repair the damage. Okay, so Ra51, it's a major player in this process, and we can study it in the test tube biochemically, but we can also visualize it in cells. And you've seen some of this before with 53BP1. So this is showing you that under non-damaging conditions, you can see that there are a small number of foci corresponding to sites of double-strand breaks that arise spontaneously, probably from collapsed replication forks. But if you treat with ionizing radiation, you induce double-strand breaks at a very high frequency, and you can see that Ra51 is recruited to the double-strand breaks where it forms these foci. Okay. So why am I telling you this? Well, again, I want to come back and just to illustrate the links between this process and human disease. Many of you will have heard of the BRCA genes. There are 2 BRCA genes, BRCA1 and BRCA2, and these are the 2 major breast and ovarian cancer tumor suppressor genes. So people who inherit a defective copy of either BRCA1 or BRCA2 have at least a 75% lifetime risk of developing breast and ovarian cancer. Why? Well, it turns out that both BRCA1 and BRCA2 are essential for this process, for homologous recombination. And in fact, if you are defective for BRCA1 or defective for BRCA2, you are unable to target the Ra51 protein to the double-strand break. And they differ slightly in how they do that. BRCA1 actually plays a role in processing the double-strand break to generate a substrate for recombination. And BRCA2 acts as what's called a mediator, and I'll tell you what that is in a minute. But essentially, both of these genes are essential for homologous recombination. And it is that is defective in these individuals with breast and ovarian cancer. So what happens if you don't have BRCA1 or BRCA2? Well, non-homologous end joining has to do the repair. And what happens is you get these translocations that you've seen, and this is, again, these harlequin chromosomes that you can see here are fusions between different chromosomes. And it is these events that drive genome instability and ultimately lead to cancer. Okay, so this is my very simple model of how recombination occurs. So if you generate a double-strand break, first of all, you need to recognize it. And the major player is a protein called the mRNA complex. What this does, essentially, is it acts as a kind of a molecular Velcro to hold the ends together. If you don't do that, the ends are going to fly apart. So this engages with the break. It's the thing that gets there first. This then recruits various other factors that help the process. One of which is this kinase, and you probably just saw that quickly happen. These essentially are like, I suppose you could call them Pac-Man. They nibble away at the ends to generate this 3' overhang, which is essentially the substrate for recombination. Well, at least this is the substrate. It's single-stranded DNA bound by replication protein 8. Okay, so what does BRCA2 do in this process? Well, BRCA2 actually binds to the Ra51 protein. It directly binds to it, and it actually brings it to the site of damage. It brings it to the break, and then it helps to promote the formation of that filament that is absolutely essential for the repair process, which is basically how this engages with the intact chromosome. This invades, as you've seen before in the videos. With a bit of magic, ultimately you repair your double-strand break. Okay, so how do we study this process? Well, as Jean mentioned, we actually use C. elegans. This is a little worm. It was really pioneered by Sidney Brenner, and you're actually seeing videos of green worms in my lab, and this is actually from the initial fertilization of the egg all the way up to a worm, and you'll see this during the course of this video. Why do we study the worm? Well, it has quite a short lifespan from life cycle. You can go from a fertilized egg all the way up to a reproductive adult in about 72 hours. The great thing is we can maintain this in the lab at room temperature. And one of the things I think is really important is we can actually freeze the worms down. We can keep these in -80, which is a very cold freezer, essentially indefinitely. We can recover these 20, 30, 40 years later, and they grow again. We have an entire cell fate map. There are 959 cells in the worm, and we know exactly where they come from. But the reason my lab studies the worm is because it's a very powerful system for genetics. We can manipulate the worm very quickly. So I'll just give you an example. We can do an experiment in 2 weeks that might take someone in a mouse, for example, a year and a half. So we can do things much quicker. So what we found, and I think this is quite important, and again, Jean alluded to this, is that the worm has certain unique features that allow us to study it, and over the last 10 years in my lab, we've actually discovered that many of the disease genes that I've been talking about are actually conserved. That is, that they are present. There are equivalents in the worm that do the same job as they do in humans, at least at the level of DNA and DNA repair. So I've mentioned the BRCA1 and BRCA2 genes. There are equivalents in the worm that do exactly what I've shown in the videos. There are other disease processes. Fanconi Anemia is a rare disorder that's involved in repairing stalled replication forks. This is conserved in the worm, and we've been able to find out new things about how this process works from studying the worm, et cetera, et cetera. So one important thing is that some of these pathways that I'm showing you here are conserved in worms. That is, that the equivalents are present in worms and also obviously in humans, But arguably the most powerful genetic system used in molecular biology is budding and fission yeast, and they're not present in those species. So this gives us an advantage. So what I'm going to give you is an example of how we use the worm and how this has provided some insights into human disease. So basically how we use the system to study this process of recombination. And it relates to how you control recombination. So this is just another way of showing what I've shown you in videos. Oops, sorry. We have a double-strand break. The ends are chopped back. One of the ends is invaded into the intact chromosome. You do synthesis to fill in the gap and to repair the damage. You then process this and you get repair. And what I'm showing you here are just some of the factors that have been implicated in this process. But you'll notice that, in fact, on this side, there's very little information. And what we were particularly interested in is how this was negatively controlled. Now, pretty much, and I think it's fair to say this, all processes in biology are both positively and negatively regulated. And at the time that we started this work in my lab, there were known negative regulators of this pathway, at least in complex organisms. I say complex organisms. It turns out that a protein had been discovered about 30 years ago in yeast called SRS2. I'm having problems with this controller. SRS2. And I'm going to briefly introduce you to this, because this really was a paradigm for what we went on to study. So what SRS2 is basically a motor protein. It moves along DNA. And in doing so, it can do various jobs. So as I mentioned, this protein was discovered in yeast over 30 years ago. And it turns out that if you knock this gene out, if you get rid of this protein, the yeast can't control recombination anymore. Recombination becomes uncontrolled. You get what's called hyperrecombination. And this is very important. This control of recombination is very important for DNA repair. And this kind of manifests in the mutants of SRS2 as DNA damage sensitivity. So how does SRS2 work? Well, it uses its ability to move along DNA to actually displace Rad51 from the filament. So it can actually use this motor activity to drive the Rad51 protein from the filament and thereby reverse, or at least prevent, recombination from occurring. So it does this. This is what SRS2 does, is it prevents those filaments from forming. Okay, now this is very important. This particular gene is very important, as I mentioned, for genome stability in yeast. But there are no obvious equivalents in more complex organisms like worms, like fruit flies, like mice, like humans. But we reasoned that there had to be something like this. So what we did was we decided to do a genetic experiment to try and find something that was equivalent. And the experiment we did was based on this following observation. It turns out that SRS2, which is this motor protein, is normally viable in yeast, but if you combine it with a mutation in another motor protein called SGS1, which actually is the yeast homologue of a human gene that is mutated in a Bloom syndrome. So if you knock both of these genes out, the yeast is dead. It's what's called synthetic lethal, because we're combining two different viable mutations, and this gives rise to lethality. Now, we know why these are dying. It turns out that they accumulate massive levels of toxic recombination intermediates. In fact, what's remarkable is that Rad51 protein that I've mentioned, this factor that promotes recombination, if you knock out Rad51 in this double mutant, the yeast now live. So if you remove recombination altogether, they live. And so based on that observation, the lethality is due to the accumulation of toxic recombination. Okay, so why am I telling you this? Well, what we did was basically to try and use the fact that we have a homologue, an equivalent of this SGS1 protein in worms. And I'm sorry about the names, but in C. elegans it's called HIM6. HIM6 is basically SGS1. So what we did was we screened for genes, mutations, that cause synthetic lethality in this background. Okay, we just simply looked for lethal genes. And cut a long story short, we discovered a gene called ARTEL. Okay? This turned out also to be a motor protein that can move along DNA. This is just showing you what happens. Here we have a normal, healthy worm, and it produces 100% viable progeny. However, when you combine both RTEL1 and HIM6 mutations, you can see this strong effect on viability. Less than 10% of the worms actually survive in any one generation. Okay, so what we went on to show is that, in fact, this mutation in RTEL is very similar, gives the same result, same problem, same defects as what is seen for SRS2 in yeast. So you see DNA damage sensitivity, you see hyper-recombination, recombination is uncontrolled, yet SRS2 and RTEL don't look at all like each other. So we extended this to human cells. We could show that some of these features were also true in human cells. And then we went on to study how this protein actually works. So this is actually how we visualize those Ra51 filaments in an experiment in the laboratory. So what you can do is to take radio-labeled single-stranded DNA. You can then add the Ra51 protein, and it binds and forms the filament. And if we run this through a gel, this species is retarded, and we can see this difference from unbound single-stranded DNA to bound by Ra51. So these are those filaments that I've been describing. What we found is that, in fact, adding the RTEL motor protein to this particular reaction had no measurable impact on the stability of these filaments. You can see that they're still there. And this is actually in stark contrast to what would happen if we added SRS2, this yeast protein, because SRS2 will strip Ra51 from these filaments, returning the DNA to single-stranded DNA here. So that was not the answer. It turns out that what RTEL does is acts at the level of this intermediate here, which you may recall is when the broken end of the molecule has invaded into the homologous duplex DNA. And this is just an experiment showing you how RTEL works. We can form this structure in a test tube with purified proteins. So we have a single-stranded DNA, and add Rad51 as we do here, and we can form this intermediate in the test tube. If we add increasing concentrations of this motor protein, RTEL, what it can do is displace the broken end of the molecule to do this. It can basically reverse recombination this way. It can basically displace the invaded strand to reverse the recombination reaction. And we believe this is how RTEL counteracts toxic recombination. So this is what it does. I've shown you that Rad51 invades into the intact duplex. What RTEL does is reverse that. So basically you get this balance between invasion and displacement. That's what RTEL is doing. It's kind of acting as an antagonist of Rad51. Okay, so what we've been doing with this is trying to understand how this activity, this ability to control recombination, is utilized in cells. And we've shown that this is important for DNA repair. We've shown that it's important in meiosis, in the establishment of gametes. And more recently, we've been looking at how this works at telomeres. And so that's what I'm going to do in the final part of the talk, is just tell you a little bit about telomeres and how RTEL controls telomere function. So as I mentioned, all linear chromosomes end in a double-strand break. So how is it that that end of the chromosome is not used as a substrate for repair? Well, these red structures, and they're not really red, this is just an illustration, are the telomeres. These are specialized structures that exist at the end of chromosomes to protect the end. So they're actually very specialized in that they're made up of a repetitive DNA sequence. This is the human sequence, TTA, GGG, and the blue and the red here represent multiple copies of that sequence. So it turns out that this repetitive sequence can be bound by specific proteins that recognize this sequence. And these group of proteins form a complex, basically a protein DNA complex, that protects the end of the chromosome. So it protects the chromosome end, but it also helps the machinery, the copying machinery that replicates the chromosome to replicate the chromosome end. So why am I telling you this? Well, the name RTEL comes from the fact that this gene was first discovered as a factor that controls telomere length differences in different vertebrate species. And it turns out that when you knock RTEL out in invertebrates, you get massive genome instability. What that means is that the chromosomes start to fall apart. We see lots of breaks in the chromosomes, but we also see a really remarkable phenotype at the telomeres, which I'm going to show you. We see telomere shortening, loss, and fragility. I mean, the telomeres are really messed up. What we wanted to know is what RTEL is actually doing at the telomere. Okay, so the way we do this is we use vertebrate cells where we can conditionally inactivate RTEL. What that means is that we can add a virus that will induce the loss of the RTEL gene in a very short period of time. This is the RTEL protein. You can see that when we add the virus, the plus here, the protein disappears very rapidly. And this allows us to study this essential gene. We have a window of opportunity to study this in these cells. Okay, so this was the cartoon of the chromosome and the telomere. This is now actually a chromosome that we're studying, and we can actually label the telomeres, which you can see in green. So this is a normal chromosome, and you can see the telomeres at the end. This is what the telomeres look like when you don't have RTEL. So this is a low magnification image, and I'm going to show you some higher magnification images here. What you see when you inactivate RTEL is that the telomeres become very short. You can see that by the fact that the intensity of the spot of the telomere is dramatically reduced. We see complete loss of the telomere in a high number of cases. Approximately about 15 to 20% of the chromosomes in any one cell completely lose the telomere. And we also see fragile telomeres. So what does that mean? Well, fragile telomeres are where we see multiple spatially distinct telomere spots of the chromosome end, as you can see here. So what I'm going to tell you is how this and this happens. And it turns out that this is a reflection of RTEL's ability to control recombination. Okay, so this coming back to this schematic of a telomere. So you've seen this. Well, it turns out that telomeres aren't always linear like this. So this was a very famous image of a telomere from human cells. And you can see it forms, this is actually an electron micrograph of a human telomere. You can see it forms a kind of a lasso-like structure, okay? How is it like this? Well, it turns out that you use recombination using this end of the telomere. It invades back on itself to form this lasso-like structure, which is called a T-loop. So you've probably seen this before. This T-loop looks just like the D-loop, which is what RTEL acts on. Okay. So what we have found is that, in fact, this activity of RTEL, which I've shown you, is RTEL can counteract the ability of Rab51 to invade the broken end of the molecule. RTEL can reverse this process. What it does at telomeres is this. It plays a role in the dismantling of this T-loop structure to linearize the chromosome to allow it to be copied correctly. Okay, so how is it that you can go from this to this? How can you lose an entire chromosome end very rapidly? Well, it turns out that this particular substrate, if it's persistent, if it continues in the absence of RTEL, so if you can't dismantle it because you don't have RTEL, then the whole telomere gets chopped off as a circle. And we can actually show that telomere circles accumulate in these RTEL cells. So when do they form, and who's cutting the DNA to produce these circles? Well, it turns out that the very act of replicating the chromosomes is linked to when this is processed as a circle. So when the replisome approaches, so this is the machinery that copies DNA, this is actually triggering the cutting of this structure to give rise to a circle. I'm just going to show you a couple of experiments. So we can actually detect telomere circles on gels, and I won't go into the details of this. This black signal here is the telomere circle when we inactivate RTEL. But if you block replication with an inhibitor, we completely abolish these circles. So you need active DNA replication, so active synthesis of the DNA in order to get these circles. We can use different inhibitors. We get the same effect, which is shown here. We can use a drug called afidacolin, which directly inhibits the machinery that replicates the DNA, or we can inhibit something else that basically eliminates the substrate for DNA synthesis. The same effect is true. So it turns out that when you block telomere circle formation, you completely block the loss of the telomere. And this is just it's a graph showing you the number of telomeres lost in a given cell. So this is what happens in normal cells. But when you inactivate RTEL, you see a lot of chromosomes that have completely lost their telomeres. If you block replication, you completely inhibit this event. Okay? So these are the only real experiments I'm going to show you. So inhibiting DNA replication prevents these circles from forming, and it suppresses the loss of the telomere as a circle. Okay, so who are the scissors? Who is the machine that is actually cutting this substrate here to give rise to a circle? So you've already seen that during the process of recombination, we use essentially enzymes, machines that actually chop up the DNA to generate that substrate. Well, it turns out that there are other specialized DNA processing enzymes and proteins in the cell that will recognize these kinds of substrates. And it turns out that the enzyme that is responsible for cleaving this telomere T-loop as a circle is this protein complex called the SLX4 complex. It has 3 enzymes that can cut DNA. And biochemical studies in the test tube have shown that this is actually a Holiday Junction resolver. Remember that specialist structure that forms during recombination? Well, this is preferentially cleaved by the activity of this complex. And the prediction is that if SLX4 cleaves this structure, you'll get a circle. Okay? And that's what we went on to show. And it turns out that this complex is actually present at telomeres. And you can see that here. This is telomeres that are coincident with the SLX4 proteins, and you can see them in yellow when they're coincident. So when we inactivate the SLX4 protein using a genetic trick, we completely eliminate the circles that form in the absence of RTEL, and we also rescue telomere loss. So this is now, in the absence of RTEL, we accumulate lots of telomeres. We lose telomeres at a very high frequency, but when you inhibit the SLX4 gene, we suppress this. Okay. So this is just a model of what I've shown you. So you form these specialist structures at the end of chromosomes, and this helps to mask the chromosome end, the double-strand break at the end, from being repaired. So what normally happens is that when you try to copy your DNA, this would normally be a problem, but what RTEL does is it comes in and it disengages the broken end to linearize the telomere. This then allows copying of the rest of the telomere, and you don't have a problem. However, if you don't have RTEL, you have a problem. You have this persistent structure, and what we've discovered is that, in fact, these specialist scissors that can act on this particular substrate cleave this particular intermediate. This forms a circle, and this accounts for the loss of the telomere in the RTEL deficient cells. So why am I telling you this? I mentioned that the starting point for all of this is that we are using C. elegans as a model to try and understand human disease. Well, it turns out that the genome instability, these effects on the telomere, the effects on chromosomes, is strongly linked to cancer. So it turns out that if you inherit a defective copy of RTEL, you have a very high risk of developing certain brain cancers, such as glioma. If you make too much of RTEL, then you have a very high likelihood of developing gastric cancers. Why? Well, our studies in the worm and in these vertebrate conditional cells have shown that this is likely to be due to a defect in controlling this process of recombination. So what I'll do is just acknowledge everybody who's been working with me over the years. This is actually all the people that have been in my lab since 2002, some of which are here down at the front. We've also worked, as is the case in science, with many people internationally, and our collaborators are listed here, and we're very grateful to them. These are the people in red who are in my lab at the moment. As I say, many of them are here, and some of them you may see a little bit later. They're here. Finally, the people who funded the work, we are indebted to the fantastic support we have from Cancer Research UK, from the European Research Council, and many other funding agencies that help to fund our work. So thank you for your attention, thank you for coming, and I'd be happy to take questions. Thank you.
Thank you very much, Simon.
Thank you, Jean.
And we will take questions now. Could I ask that you put up your hands if you want to ask a question? We have 2 microphones, one each side of the room, and if you could wait until the microphone arrives before speaking, otherwise the question can't be heard. So, first question. Still thinking about it?
So, if I understood you correctly, you can RTEL can be you can either have too much of this enzyme or too little, and depending on that, you get different sorts of cancer.
Correct.
So, does that explain all types of cancer?
No, I mean, it's a great question. I mean, one of the things that we really don't understand with respect to cancer is why certain mutations in certain genes give rise to a very specific kind of cancer. It's really not well understood why that is. I mean, obviously I'm giving you as RTEL as an example, but there are many other what are called tumor suppressor genes or oncogenes. So tumor suppressor genes are obviously the mutations that when you inactivate them, you have an increased cancer risk, whereas an oncogene is a gene when you overexpress, overproduce it, that is actually driving cancer. Now, for whatever reason, specific mutations in specific genes can give rise to cancers in very different tissues, and that's something we really don't understand very well at all. Why the mutations in BRCA1 and BRCA2, these breast cancer tumor suppressor genes, why do they predispose to breast and ovarian cancer? We really don't know. Really don't know. It's a great question. It's something we don't understand.
One behind you.
This is fascinating findings. Just purely an aside, really. You mentioned using a viral mediator to inactivate the RTEL.
Yes.
Is there any risks using such an approach?
Yeah, of course. So I didn't go into the specifics, but it is the virus we use expresses a recombinase that actually recombines out part of the RTEL gene to inactivate it. So the risk that we use, I mean, we're obviously doing this transiently in cell culture in the laboratory, but this recombinase can actually act promiscuously on other sequences in the DNA and can cause a few problems. So we are aware of that and we can control for that. But a lot of, I think maybe if your question is more kind of generic about the use of viruses, they're used extensively in the laboratory to allow us to do various things to make proteins, or in this particular case, to make a recombinase to allow us to inactivate the gene. There are risks because they're virus, but these are extremely attenuated viruses. They're not pathogenic, they don't cause problems to us. But there are some issues that we have with the cells that we kind of have to control for. Yeah.
If we are lucky enough that all our repair mechanisms work,
Yes.
does that mean we can expose ourselves to lots of mutagens and not worry?
Well, nothing is perfect. The way we copy our DNA is not absolutely perfect. I mean, unfortunately, we age, and aging is a manifestation of the accumulation of mutations in our DNA. And you can see that certain people age faster than others. Some people are cancer predisposed, and others are not. But I think if you were to live long enough without cancer, eventually you would get cancer, just because I think it's part of the process. You will accumulate changes in your DNA over time. The repair mechanisms are very, very good at repairing DNA damage, but sometimes they make mistakes. It's not completely fail-safe, is what I'm saying. So even if you have a perfect repertoire of all of the repair machinery in your cells, and some people probably do have, and others may have ones that maybe don't work quite as effectively, and you've seen some extreme cases where some people are completely defective for a particular repair process, then you're going to accumulate problems. So I don't think you could have a completely perfect repertoire of DNA repair and kind of never get cancer, because the problem is that these machines aren't absolutely fail-safe. They don't always get it right. They don't always get there and repair the damage correctly. I hope that answers your question.
Hi, I was just wondering, do you think this knowledge about how RTEL works will sort of help cancer therapy in the near future?
It's a good question. I think it's very early days. I think this is quite a new area that we're embarking on. As I kind of mentioned to the other gentleman, there are many mutations in many genes that can give rise to cancer. This is just one more of those, but certainly one way that we could potentially help at this point is identify high-risk groups, people who are more likely to have mutations in this particular gene based on the fact that they have a family predisposition to the kind of cancers that we're talking about. And you could then screen individuals for mutations in the gene, and that would then give you an idea of whether or not that person might be at risk. So that is something that you could, in theory, do right now. In terms of treatments, well, it's very early days. I mean, this is the premise for basic research into this kind of topic is to develop a platform of understanding that hopefully will allow us to develop targeted therapies. And we're very early on the road with this particular project. I mean, the BRCA genes I mentioned to you, and this is a great example of where we hope maybe RTEL research will go. So there are in fact now therapies that are showing great promise based on the knowledge that has been gained from studying the BRCA genes. And we're hopeful that this is going to work out. But in terms of RTEL and what we've found, it's very early days. But, yeah.
Any further questions, Lurking? No, I think we're probably finished there.
Right.
So it remains for me now to thank Simon for an excellent talk. I think Francis Crick and Watson, just about 60 years ago, that they announced the discovery of the DNA double helix. And I think Francis Crick would have been fascinated to hear this talk and see the level of understanding and detail that questions are being addressed now in this important field. So I think Simon's an excellent choice for the lecture. Somewhere I have to give him some things. So again, congratulations on the award. And here's your scroll.
Thank you.
Here's the medal.
Wonderful. Thank you.
And the important check.
Thank you.
Okay.
Thank you very much. Thank you.